In this video, we're going to talk about male reproductive system, and first start with meiosis, and then we'll get to the female reproductive system later. This is looking at the functions of both reproductive systems. So developmental hormones, meaning development of offspring, are going to be released. That's going to help gestation of offspring. So this is actually in utero. So this is going to happen in the female. Fertilization is going to be sperm and egg coming together to create offspring. So ultimately, the goal of the reproductive system is for reproduction, for offspring. We also release androgen and androgens are going to help with kind of female and male characteristics that we see. In the reproductive system, and they're helping with gamete production. So we'll talk about those a lot more in each one, whether it's male or female. First, we're going to talk about meiosis. Meiosis is only happening in the gametes. So mitosis happens everywhere else where you're just making exact copies of your cells. In the sperm and the eggs or the ova, we're making kind of individual copies. So we're going to take your genetic material, kind of scramble it together and make individuals. That's why you're not a direct copy from your mom and dad. We get different combinations of maternal and paternal genetic material. So we're going to look at that. So in mitosis, we have just direct copies, and we have 23 pairs of chromosomes because one comes from female side and one comes from male side. So maternal and paternal chromosomes. So we have pairs. So we'll kind of line them up that we say chromosome 1, and then 2, 3. So we'll have kind of 2 chromosomes there. One's from maternal side, so mom, and one's from paternal side, from dad. So we have kind of this combination going on all of them up to 23. When we do meiosis, we actually halve the number of chromosomes because an egg and a sperm come together to make 46, to have 23 pairs. So we only want 23 chromosomes in each the egg and the sperm. So when they come together, they make 46. Meiosis, we're going to see that has 2 phases. There's meiosis 1 and meiosis 2. So you're actually going to see where it'll go make 2 new cells, and then those are going to divide. So ultimately, we're going to have 4 cells produced when we go through meiosis. So looking at meiosis, this is the same as mitosis. During interphase, we replicate all of our chromosomes. So that's going to get us ready for the division. During meiosis 1, we have what we call homologous pairs are going to separate from each other. So again, we're going to copy it. So we're going to have a pair that is maternal genetic material, and we're going to have a pair that's paternal genetic material. Then those guys are going to separate. And we say it's halving the number of chromosomes because we kind of consider this one chromosome, even though it's 2 chromatids together. So we consider it kind of halving the amount of genetic material in meiosis 1. And then during meiosis 2, we're going to take those pairs and separate each individual chromatid from each other. And they'll each go into the final cell so that we have cells that are ready to be fertilized. So whether it's an egg or a sperm. So looking again at meiosis 1, which is going to be where different things are going to happen from mitosis. Meiosis 2 is pretty much mitosis again, except we're only dealing with homologous pairs, and we're dividing the amount of chromosomes down. So in prophase 1, we see a couple of things. Homologous pairs actually line up to where we have four chromatids, so the four single lines that I show you. So say this is chromosome 1. We're going to have two copies of our maternal genetic material, and we're going to have two copies of paternal. And same thing. So these guys are all going to hang out. And then so chromosome 2, same thing. We're going to have a line of 4. During this time, we actually have what we call crossover, where a piece of maternal genetic material can actually go over to paternal, and then we swap them. So little pieces can swap between maternal and paternal chromatids, and so that we're getting new combinations. So this chromosome is different from what you started because we swapped some genetic material. Why do we do this? Genetic diversity. So again, you're not an exact clone of either one of your parents, maternal or paternal side. So we are unique and individual because partly this, where we combine our genetic material. Also, during meiosis 1, remember metaphase is where all is lining up. So a lot of the phases are happening the same that we learned in mitosis. But meiosis 1, part of kind of the randomness and the new combinations is what we call the tetrads, which is again the copies of maternal and paternal with their new kind of combinations can line up randomly. So this one's going to go to this side, this one's going to go to this side, but then we can have paternal on this side and maternal on this side, so they can kind of switch places. They can line up randomly. And so that leads to kind of random assortment and mixing, again, more mixing of the genetic material. During anaphase 1, again, sister chromatids of one of the chromosomes. So you're going to have separation like this, where two copies are going to go to this side and two copies are going to go to this side. Now remember with mitosis, we just had them all line up in the middle with their copies, and then the separate copies kind of moved to each side. And so we had 46 chromosomes line up in the middle, so we get or 46 pairs of chromosomes line up in the middle, and then we get 46 on this side and 46 on this side. This way, we're just lining them up where half are going to this side and half are going to that side. So we're not making sure that they're all lined up together. So we're getting, again, half the amount of chromosomes because eventually we want to get to 23 chromosomes, not 46. So if we're looking at meiosis 1, we have two copies of one member, meaning we either have a cell with maternal or we have a cell with paternal. They've separated. We still have to, again, separate. We say that we've half the number of chromosomes, even though we still technically will have kind of united and copied chromosomes. The fact that we're going to divide again, we're going to divide. Basically, meiosis 2 is another mitosis, where eventually we're going to have that and that is going to separate, and then one of each from here too. So again, haploid is going to be half the number of chromosomes. So eventually we're going to get to it where there's just one per cell, if you look at it that way. So again, this is looking at mitosis. We start with the chromosomes. We have all of our pairs when we have duplication, but we're going to have one kind of set go to one side and one set go to the other side. So again, they're going to be exact copies of each other. In meiosis, we're going to have meiosis 1, where we have the tetrads. We have four. And we're also having the crossing over. So these should actually have new, like, little red section here, and it'll be blue section there. So we can have combination. But what happens is, see how this cell has a black pair and this cell has the gray pair. Same thing, red, orange, kind of darker blue, lighter blue. So eventually cells are going to have kind of copies where we just separated them. The difference between mitosis where we just have the one singular ones. Then meiosis 2 is pretty much like mitosis where we're just separating these again. So we have one black here and one black here, and then gray here, gray here. So we separate them all into individual cells, and we have four. So the major part is we have genetic diversity, and we have half the number of chromosomes. Because again, this is happening for both sperm and eggs. And we'll talk about kind of where it happens in both. But the important is a sperm's going to have 23 chromosomes and an egg's going to have 23 chromosomes. They're going to come together, make 46, and be a brand new combination. So looking at sperm, and this is looking at some of the anatomical features of the male reproductive tract, sperm is produced in the testes, and we'll talk about the production of sperm later. But then it travels into what we call the epididymis. So this is going to be the epididymis here. It's got a farther head area and then goes down to a tail. So this is kind of sitting on the surface of each testes. There's two of them. And then we travel actually from the outside of the body. This is in the scrotum, so outside the body, to go inside to kind of wrap around and then come back out. So sperm is going to travel around the ductus deferens. You might have heard it as the vas deferens. We call it the ductus deferens now. So the ductus deferens goes inside the body, wraps around the superior portion of the bladder, comes back around the posterior side, and comes back down. Then it's going to travel actually in the urethra. So the urethra is actually for males, both for urine and for reproduction. So it's for both, and we'll talk about how it kind of changes based on the need. We also have what we call an ejaculatory duct. That's going to be right here. It's going to be a combination of the seminal glands, which we'll talk about, coming together with secretions and then going into the urethra. So we're going to talk about all of these, but that's the path of sperm that's going to happen. And talk about the accessory glands. They're just adding fluid to sperm to make semen, so helping sperm make their travels and survive. So seminal glands, the prostate, and the bulbourethral glands are going to secrete fluids into, again, the ejaculatory duct or the urethra, and it's going to combine with sperm to make semen. So the urethra, again, is going to go through the penis and exit the body. It is for the urinary tract, but it's also for reproductive tract in males, not in females. So again, we're going to look at the seminal glands, which are going to be on the posterior side of the bladder. The prostate actually surrounds the urethra. So you can see right here is the urethra coming from the urinary bladder right here and going through the penis. And then we have this little guy right here, which is the bulbourethral gland. And that's going to add secretions as well. So we have kind of multiple places where we're adding fluid from those glands that, again, are adding to the sperm. And we'll talk about the function of each one later. So looking at the testes in utero, so in the uterus, testes kind of form, and they're forming in the abdomen. So they actually form in the abdominal cavity, just kind of around the kidneys. So we have a connective tissue band that connects to the testy and connects to the scrotum or down near the scrotum. What happens is the connective tissue band stays kind of the same size, but the fetus is growing. So that pulls the testes down towards the scrotum. Eventually, the connective tissue can contract, and it actually pulls the testes through the inguinal canal into the scrotum where they are at birth. So they pull actually out through the abdominal wall into the scrotum. So they're actually produced in the abdomen and then pulled out, and this is happening in utero. This isn't kind of important. Sometimes it can be where testes are left behind and they don't actually meet into the scrotum. So there is kind of issues where testes will stay in the abdomen, and that's not good because testes like temperature control, and that can't happen in the abdomen. It can only happen in the scrotum. So they can actually cause issues where they can become cancerous and have to be removed. So if we look at the scrotal cavities, the scrotum is going to be where the testes are staying. So we have kind of sac of skin outside, and then there's two testes, testicle. And we also have what we call a scrotal septum, which separates the two. So you have one half for one testy and one half for the other. So we can also see kind of the scrotal cavity separation. There's a little kind of thickened raised area that's on the surface of the scrotum called the raphe of the scrotum. That's going to show the separation between the two sides. We also have two smooth muscles, the dartos muscle and the cremaster muscle, that are involved in temperature control. So the dartos muscle is actually in the surface of the skin of the scrotum. So it's actually kind of all along the surface of the scrotum here. Why is that there? Well, when the testicles are kind of cold, when male is cold, it can actually wrinkle the skin and pull them up towards the body. So it pulls the testicles up towards the body where there's more heat. So it can use body heat to warm it up and then get back to the normal temperature. So again, testes are very specific on the type of temperature that they want to be maintained at. So they have kind of ways where they can relax and get away from the body and try to cool down, or come towards the body to warm up. The cremaster muscle is going to be kind of running along the spermatic cord. So going down to each of the testes. And see that later. So we're going to see cremaster muscle kind of running along here. This is another way where we can bring the testes closer to the body. It can happen both during sexual arousal, but also cold temperatures, same. So we have the dartos muscle bringing the scrotum closer. We also have the cremaster muscles, which is going to contract and can pull the testes closer to the body. Again, colder temperatures felt in the scrotum is going to bring the testes closer, so it can use increased body heat to increase the temperatures in the testes. So looking again, the spermatic cords are going to be going through the inguinal canals, which is going to be a hole in your abdominal muscles. That's going to be in the groin area, basically. And that's going to be kind of connection between the abdominal pelvic cavity, so inside, and the testes, which are on the outside in the scrotum. So basically, those are going to be where all of the connection is between blood vessels. The cremaster muscle is going to be there. Nerves, so sensation, lymphatic vessels, all need to travel to the testes. So we need communication between the testes and the rest of the body. And that's happening through the spermatic cord. So the spermatic cord is going to contain the ductus deferens, blood vessels, nerves, all of those things. So connection between the rest of the body, and you can say inguinal canal right here, and then goes into the abdominal pelvic cavity. We say the entrance to the canal, so there is a little kind of section canal part that we say if it's on the exterior, the first, if you're going from the testy into the abdominal pelvic cavity, the first thing you go through when you hit the inguinal canal is called the superficial inguinal ring. So again, the inguinal canal is going to be from the superficial inguinal ring to the scrotal cavity. So we'll see kind of a long kind of channel that you see. So it goes through again, going to, so you can have exit for the spermatic cord for sperm. And you'll see that a lot more in lab as well. What happens is this is a weakened spot for the abdominal muscles or the abdominal wall. And so this is where inguinal hernias can actually occur, where visceral tissue, so fat or visceral organs, can actually go and leave the abdominal cavity and go into the spermatic cord and even go into the scrotum. So this actually goes through the inguinal canal and causes issues. So this is a surgical fix where it had to kind of add mesh to kind of tighten up the inguinal canal and make it a better hole or kind of a stronger part of the abdominal wall so that we can't have abdominal organs coming out. So this is common in males. If you've heard of an inguinal hernia, that's going to be where it's occurring. So looking at it again, we have the superficial inguinal ring. So again, kind of a hole in the abdominal wall. This is showing you the cremaster muscle that's covering. Dartos muscle is going to be again in the skin surface of the scrotum. This is the raphe, or again, separating. We have septum that's going to separate. The raph's right here. Separate two sides, just two testes. And then we also have, this is kind of taking off the cremaster and all the fascia and looking at the spermatic cord and all that's kind of contained. We have the vas deferens, we have nerves, we have arteries. We also have a venous plexus, and we'll talk about that too. That's the piniform plexus. That's going to help with cooling as well, because we need to make sure this is all temperature controlled. So this is looking again at spermatic cord. And then we have the inguinal canal, which is going to be just kind of this starts at the superficial ring, kind of going through. So we have inguinal canal going through the abdominal wall and then into the abdomen. So I mentioned, so blood supplies, we've learned this in lab that the testicular arteries are going to be coming down from the abdominal aorta. The testicular veins are going to come out from what we call the piniform venous plexus. And that's going to be surrounding each testicular artery. So you can see, saw it here. So you can see this is the testicular artery coming down. This is the piniform plexus here, and it leads to the testicular vein. Why is the piniform plexus surrounding each testicular artery? It's to cool down the testicles. Again, keep the testes cool. It's actually going to take heat from the testicular arteries that are coming from the abdominal aorta. And because remember, veins are going back towards the body. So the venous supply has been out in the scrotum and the testes for longer. So the arteries are coming in and the veins are going back. So the veins have time, the blood in the vein side has time to cool down to temperatures um, similar to the testes. The arteries are coming off abdominal aorta, they are going to be body temperature. And we don't want body temperature in the scrotum. We want lower than that. So the testicular vein is going to help, or the pampiniform plexus is going to help cool off the blood so that, again, when it's coming down into the testes, it's cooler. We also have two kind of membranes that we have surrounding kind of the scrotum and the testes. So the tunica vaginalis, I think it's named more based on females than males, is going to be another serous membrane, so our famous serous membranes, that's actually going to line the scrotal cavity. So again, the way the scrotum is, we have the scrotum septum, we have testes here, and we have testes here. So the tunica vaginalis is going to be on the surface, line the scrotal cavity, and it's going to, again, serous membrane makes fluid, so it reduces friction. We've seen this time and time again. Because there's not direct connection and there can be movement of the scrotum and the testes separately, so we don't want them kind of rubbing against each other and causing damage. So we need to reduce the friction. On the tunica albuginea is going to be a fibrous capsule that's going to cover the actual surface of each testis. It can also go on the inside, that's going to divide the testes into specific lobules, which we'll see with sperm production. So it's actually going to divide, when you look in the inside of the testicle, into sections or lobules. So we'll see that. Then in the inside of each testis, we have what we call seminiferous tubules. This is going to be where sperm is actually produced. It matures as it's going along, but it's produced in the seminiferous tubules. And they're coiled kind of within the lobules. There's a lot of surface area for sperm production. There's also what we call myoid cells. So remember, MYO is muscle. So these, spelling MYO again, muscle cells. These are actually contractile cells that can help move sperm through the tubule. So as it's produced in each step that goes along, it needs to be transported. So then once it's ready, it's in the right place. We also have the ret testes, which is going to be where the seminiferous tubules kind of all combine. So there's multiple seminiferous tubules. They lead into a straight tubule, so there's kind of a straight end. And then they lead into the ret testes. So again, it's just a pathway that we're going to go on. Sperm's produced, it's continuing to mature, and then it needs eventually to get to the epididymis, so then it can go to the ductus deferens and it can exit the body. So again, we like to use efferent or afferent. So efferent ductules that are going to be in the ret testes. So we're going to see again, ret testes are going to lead to the epididymis. So it goes seminiferous tubules, straight tubules, ret testes to the epididymis. So we have efferent. Remember, E is for exit. So that's going to lead, again, to the epididymis, which is going to lead to the ductus deferens. So this is just a better picture of looking at it. So we have the tunica vaginalis, which is going to be kind of the surrounding, again, in the scrotal cavity. So again, reducing friction, serous membrane. Then we also have the tunica albuginea, fibrous capsule protecting the testes that's going to separate into lobules. So you can see all these kind of coiled tubes is your seminiferous tubules. Each one leads to a straight tubule at the end. So you can see all these guys are a little straight. That leads to your ret testes, which is this kind of network of tubes that are going to lead again to efferent ductules, which are over here. That's leading to the head of the epididymis. So head of the epididymis is the widest part. And then it's going to go down to the body, which is kind of the middle part. And then it's going to lead to the tail, which is going to make the U-turn that goes into the ductus deferens. So again, sperm are produced at the seminiferous tubules, but they mature in the epididymis. So again, sperm are produced, so they're made, but they need to mature. When they're made, they're just cells. Well, we kind of know sperm are going to have the head, a midpiece, and a tail, so they can swim. So the maturing is going to happen in the epididymis, where its cell goes to swimming sperm. Again, the epididymis has a head where the efferent ductules of the ret testes are going to empty into that wider part of the epididymis. And then we have the body, which is the middle, and the tail, which is making that U-turn that leads into the ductus deferens. Inside the epididymis, the epithelial cells are pseudostratified. So remember, it looks like there's multiple layers, but it's not. And we also have what we call microvilli, or in this sense, it's sterocilia. We have, again, wherever we're having microvilli, it's increased surface area. So this is going to have where we can, any excess testicular fluid that's added for kind of making sperm, can be absorbed, but we can also release nutrients to sperm that's stored. So the sperm is sitting there and waiting. It can be stored and held. So we need to have nourishment for it because it is a living cell. So we need to make sure that it's getting everything that it needs. And so that's where this is going to happen, as at the epithelial layer. So again, looking at the epididymis, we have the seminiferous tubules and the lobules here. And then they lead to the straight tubule, which leads to the ret testes, which is this kind of combination here. Leads to the efferent ductules, which is going to lead to the head of the epididymis. So again, the head of the epididymis is going to receive kind of the immature spermatozoa, and we can go over all of that later, from the efferent ductules, and then goes to the body and of the epididymis, and then goes to the tail, and then leads to the ductus deferens going out to the urethra. Again, non-motile sperm, so ones that can't swim, are going to go and enter the epididymis. As they're traveling through the epididymis, they start to transition from, again, a cell to where they can actually swim. So it goes from this process, and it takes about 20 days from going from cell to actually sperm that we can see swim. They can actually be stored for several months in the epididymis. There's going to be all of that kind of surface area where kind of maturation can happen, but also storage can happen as well. During ejaculation, where sperm is released from the body into the urethra and then out, the epididymis actually contracts and helps push sperm that's ready. It's going to be more closer to the tail into the ductus deferens that will then travel into the urethra to be expelled from the body.